The overall goal of this high-content microscopy-based method is to simultaneously quantify mitochondrial morphofunction and cellular levels of reactive oxygen species, so as to establish a robust redox fingerprint for cell lines exposed to different experimental perturbations.
This method can help answer key questions in redox biology, such as whether pathogenic conditions or compound treatments elicit oxidative stress, and to what extent mitochondrial form and function are affected.
The main advantage of this technique is that all parameters can be measured within the same cell at the same time.
To start the procedure, seed 8 to 10,000 human dermal fibroblasts in 60 inner wells of a black 96-well plate with a thin continuous polystyrene or glass bottom. When using different conditions, treatments, or cell lines, distribute their seeding locations homogeneously on the plate to minimize the plate effects. Then, seed a further 8 to 10,000 cells in well B01 to be used for focus adjustment. Subsequently, fill the empty outer wells with medium to minimize the gradients between the wells and the environment. Gently tap the plate 3 times before placing it back into the incubator to avoid the cells from growing in patches. Culture the cells for 24 hours, or until they reach 70% confluency. Afterward, save the treatment information for the experiment into a spreadsheet called Setup. To set up the microscope, first calibrate the XY stage using the software. Next, create an imaging protocol for